We will start with the second part of the session, advancing pandemic preparedness across Europe at the initiative of small countries. And I would like to invite the next speaker, Dr. Marion Koopmans, Director of Department of Viroscience at Erasmus Medical Center in Netherlands, the WHO Collaborating Center for Emerging Infectious Diseases, Scientific Director for Emerging Infectious Diseases in Netherlands, Center of One Health, and Scientific Director of Pandemic and Disaster Preparedness Center in Rotterdam and Delft. She will give us a talk focusing on integration of big data, metagenetic, and citizen science for emerging disease surveillance.
Yes, thank you. And so glad you're back. And thanks for the invitation. And I would like to just share a bit of how we are maybe even scratching the surface of thinking about innovative ways of doing surveillance. Because the way I look at emerging disease preparedness and all the efforts out there is that we really are heavily focused on response. You know, understanding what to do once people are getting sick. And our lab also is very much invested in that with flu research, with coronavirus research, but we're also trying to move a little bit more with the focus on pre-emergence. Can we actually get better at predicting where these things are popping up and also at being really prepared? So, and this is work that is ongoing in a European consortium, VIO, that was mentioned this morning by Marcel Salaté. He's part of it, and the EBI is part of it. So we're really with multiple institutes on data analytics, infrastructure, and virology and ecology are trying to figure this out. So the core question is, can the risk of outbreaks or emerging diseases be predicted so that we can really target our early warning, surveillance, and prevention efforts, and also how do we bring in the novel tools like the ones that Linfa also mentioned into this picture and what we can learn from that. So we work along what we call disease emergence pathways with models for zoonotic wildlife-borne disease emergence. Think of flu, of course, vector-borne disease, a climate change scenario that focuses on what's going on in a very rapidly changing environment in Greenland, a silent epidemic scenario, I will mention some of that, and then of course, how do you bring everything that you learn and the tools that you develop together in an integrated disease X scenario. And I will just give you a feel for what we're trying to do. It is by no way, it's really exploratory with these three examples here. So, every work with any scenario puts together a group of experts from different disciplines to just collect the information, as just was mentioned in the previous talk on biosurveillance. What are key drivers for this particular scenario? What do we know about that? Which types of data could help us inform about those drivers? Think about population data, think about citizen science collected data. How good is that data? How real time, and can we really access it? And what are key knowledge gaps that we really need to fill? And then you make these very complex images, maps here of data types that we think need to be considered when moving in this direction. This is put together for looking at West Nile. So there are some elements in those boxes that you will recognize. Of course, abundance and the range of vectors is important. The same abundance and the range of hosts, bird host, if you think of West Nile. And each of these boxes is a type of driver which has its own data that can be collected through whatever means, from routine surveillance, from targeted studies, by citizens, what have you. So, and then we try to see what do you see if you bring data together. So here's an initial exploration, compiling whatever molecular data could be found across Europe from testing of birds, mosquitoes, humans, environmental samples on West Nile. And this data is usually not collected and aggregated, but if you do that, you actually see that for the dominant West Nile lineage in Europe, you see two subclusters that move quite differently if you look at this movie here. So one that moves southward, one that moves northward, and both seem to originate from this part of Europe where we have virtually no surveillance data if you look on the ECDC maps, for instance. So then the next step is to say, okay, if we see that direction of movement, can we explain that? Can we explain that if we bring in those other types of data? Here, a list that was used in this particular study. And then what do you see? So in here, what the conclusion was, well, the spread of these viruses is really directed by degree of urbanization, coverage of wetlands, presence of birds, resident birds, and migratory birds. And that, of course, is very intuitive, but it's very good. It creates confidence seeing that coming out of this analysis. It also showed spread is accelerated by agricultural intensity. So now we are moving to a country that doesn't have West Nile problems and trying to say, okay, can we use this to help us direct where we should stick our thermometer for early warning surveillance? So this is the Netherlands. It's Delta, as you know, and we have big initiatives to get our country climate ready, where we see sea level rise and where landscape architects are designing wetlands all over the place, which of course is very interesting, but is also potentially a risk for novel arbovirus disease emergence. So guided by our strategy, we then started to develop some integrated sampling through citizens, schools, and through bird ringers, and indeed found pockets of West Nile present, with adding in serology, seeing that this is there from 1 year to the next, so now 3 years, 4 years in a row, even though we don't see human cases. First year we've seen a few human cases, but that's it. We have enzootic presence of this virus that we know is a human pathogen. And the next step now is trying to see, okay, which pieces of the driver puzzle do we need to help us say, okay, when we see this happening, minimum night temperature so long, or other changes, that is when we really need to start to be careful about human disease. This is an example. Second example is trying to explore what we call the silent epidemic scenario, and that's inspired here by, this is an image from a really interesting paper that made a reconstruction of how did Ebola spread in West Africa, using the virus genomics and some metadata to see that really the big nodes of dissemination of this virus were the high-density urban slums, where really the typical control that you need was not possible. And that is illustrative of a new disease risk. We've seen the transition from very rural, localized Ebola outbreaks to this type of pattern, where things can go under the radar if that enters high-density urban regions. So what we've been exploring is what can we do with wastewater. So this is well before the pandemic, and it was put on a sidetrack for a little while because of the pandemic. But we collect wastewater samples at some regular intervals here in 63 cities, 48 countries, 6 continents, and some of them also send longitudinal samples. And then we do look at what do we find? Not one specific virus or a handful of viruses, just the entire virome. Thinking that, okay, if we want to know what is unusual, you have to first explore what is usual. What's the normal urban virome? Is it anything different? Is it different in different regions? Things like that. So we do virome sequencing, unbiased and captured. We also split the samples, and they are DNA sequenced for resistome and for bacterial population in Copenhagen in our collaborators. And then we just explore what can we learn from this? What does an urban virome tell us? Here's one of the analysis. This looks at all the viral data, just agnostic of what it is, where you can see that you have central cores where both in the non-capture and in the capture data, which are enriched for vertebrate viruses, you see there's a core of viral data that is present in all sites, but there's also outliers. And of course, then it becomes interesting to see what defines the outliers. Is that something unusual, or is that specific for a different region in the world, for instance? And here's an example, zooming in on one of the core, the known pathogens, enterovirus C. So here now, simultaneous snapshot in different parts of the world. In Quito, here in North America and in Africa, where you see there are some similarities here, Coxsackie A19, I think, circulating everywhere, but then here an epidemic wave of one enterovirus, here an epidemic wave of a different enterovirus, and here nothing standing out really, but much greater diversity. So just one virus with very different patterns in different regions. And of course this is initial data, but it is, we are now looking at in the longitudinal how stable is this information. Here's another example. This is for a norovirus, one of the diarrheal viruses. One genotype, doesn't really matter why, but there's a separate branch here only found in African region. And when then taking that virus, expressing its protein, looking at receptor binding profiles, it's a virus that behaves very differently from all the other known noroviruses. And this is, of course, then interesting to explore because we have virtually no surveillance data from a big part of the African region. So that's where we're now heading with this. And then a last example, and that's really thinking about disease X scenario, and that is how can we bring in these tools to get a deeper understanding of what is going on. And this was done on the back of the Origins mission of SARS-CoV-2. Here this is a map or an image that I took at the market, Huanan seafood market, when I was visiting in 2021. And here is the clinician who I think deserves big credit because she picked up the phone to the Wuhan CDC saying, I have some cases that that have a weird pneumonia that I haven't seen before. So she really started, I think, what we now know about these viruses. So what happened, so the basis here is the data gathered through the WHO Origins missions. That was a series of studies really to try and get at starting in Wuhan, how much earlier has this virus been circulating? What can we learn from a whole series of studies? And those were done and they were written up, but the judgment was already out. The jury was already out before we had finished our report that it was not good enough. So that was a bit a weird experience. But key findings there were that circulation did not start much earlier than December 2019. There was this complex market which seemed to have something to do with it, with links through supply chains across and beyond China. There was at that moment no documented evidence of live animal presence, but of course that was something that was very doubtful. And we considered the zoonotic origin most likely, but other scenarios were not ruled out. And we did recommend further studies to dig further back and dive deeper. Now, since then, of course, we have seen some new evidence pop up. One publication that really astonished me in July 2021 that already had been in the works for maybe a year, and that really had the documentation that there were live animals on the Huanan seafood market until November 2019, when that particular study stopped. And those were live animals, wild-caught and farmed wild animals of 38 species, including several species that we know are susceptible. In addition, what was added was work by the China CDC that had done environmental sampling and published its test results, so SARS-CoV-2 testing by PCR of environmental samples. And the data was combined by, in an initiative that I was invited to by Mike Warby, who then said, okay, if you really combine this, the rate of positivity is very much highest in this corner of the market, which also happened to be the corner where the animals were. Our next piece of information that came out was looking at the metagenomes of those same samples, where an initial preprint by China CDC concluded, well, there was mostly human DNA, but then that data was reanalyzed after it was put online. Involuntary is what we heard. But it was reanalyzed, and that showed that, yes, there was human DNA, but there certainly was also DNA of many of these different animals that had been found in that one particular survey. So definitely documentation of presence of animals or animal products, but then also of animal viruses, building further and further the case of actual animal presence. So this is without the documentation at the time from the public health system. You can still go very far in saying, okay, well, explain this then. So that's just very, very much a high-level flyover of the types of innovative surveillance approaches that we are exploring. And here I want to end with this figure from an OLAP panel report. OLAP is the One Health high-level expert panel that advises the quadripartite about where to go. And as I mentioned before, I do think One Health is really a key focus, and particularly also trying to move towards prevention, as long as we're not that good at stopping epidemics in their tracks. And of course, I have a big team to acknowledge, a wonderful group of people, a pleasure to work with, also the European collaborative, and of course, funders that make this possible.
Thank you. Our next speaker is Dr. Jochen Neitz. Dr. Neitz is coming from the University of Leuven in Belgium. He's working as a full professor of virology at the medical school and School of Dentistry. His laboratory has long-extending experience in development of antiviral strategies and drugs against emerging and neglected viral infections. Today, Dr. Neitz will give us more information about broader spectrum antivirals for epidemic and pandemic preparedness.
Thank you so much, and thank you for organizing this fantastic meeting in blue sky outside. So first of all, indeed I would like to explain a little bit what our view is on the need for development of broader spectrum antivirals. But before doing that, I think we should thank Christian for all what you have been doing. It's amazing, and I admire what you do, and I think with me, likely everybody here in the audience. Yeah. So what do we have in terms of antivirals? We have good drugs against herpes viruses, HIV, fantastic, 1 pill a day, and people live a healthy life. We have some drugs against flu, but we need better drugs against flu. Hepatitis B and C, and now even D, so you can even cure hepatitis C in 8 weeks. With 1 or 2 pills a day. We have now inhibitors for SARS-CoV-2 and even some pox virus inhibitors, but we don't have drugs against any other viral infection. And we talk all the time about epidemic and pandemic preparedness, but we still have many infections, mostly tropical infections as well, for which we don't have anything. So there is still a lot to be done, and that's what I would like to briefly explain to you. And by the way, I was on the 20th of September 2019, I was in Wuhan explaining what I'm just explaining now, that the world is not ready for the next... and Marianne Steven van Gugh tells me that that I brought... So I was explaining that the world is not ready and that we should... so if a new virus is emerging, that it will take time before we can develop a vaccine. It's not always a given that you will be able to develop a vaccine. Just think about HIV and hepatitis C and adenoviruses and herpes simplex type 1 and CMV and so many others. So it's not a given that you will be able to do it. And that you should have, in the meantime, antiviral drugs that you can deploy, because we know that each virus that will emerge one day, disease X, it will belong to a certain family, and we know all these families. And so just imagine that we had on the 10th of January 2020, when the sequence was put online of SARS-CoV-2, that the WHO had a stockpile of coronavirus inhibitors. There was remdesivir, but remdesivir had to be given intravenously, but that we had something that you could take orally, and that could have been shipped to Wuhan on the 11th of January. They could have started right away clinical studies, and it would have shown with a Paxlovid type of molecule, for example, that would have been effective therapeutically, prophylactically. And just imagine also all these people in nursing homes here in Europe that died because of the virus that was in the facility. Just imagine that we had something given prophylactically or therapeutically. So that was something, not in so much detail, but I was explaining in Wuhan. But also in 2013 we published, and this was not just in Wuhan, that it's probable that novel, potentially highly pathogenic viruses will emerge, and that we may need broader spectrum antivirals to contain such problems. And so what I'm explaining to lay people is that viruses belong indeed to a family, and before 2020 we knew 6 human coronaviruses, then came the 7th kit on the block, so we could have developed pan-corona inhibitors based on these known human viruses, and that we need to do so in the future for other families of viruses. For example, the paramyxo and the pneumoviruses. For example, we know that measles and RSV, that they can be highly infectious, and we know that the henipaviruses can be problematic, that bats are full of henipaviruses. So a paramyxo compound, and not just one compound, at least 2 with a different mechanism of action, so that you don't run into resistance problems. But we have all these other families, so there is quite some work to be done, we believe, in developing such strategies. And not only virologists said that we should be prepared. Bill Gates in a TED Talk in 2015, was it, said that we're not prepared, and in the meantime, I think 17 million people died. This was 2020, 21, 15 million. And we had the luck to welcome Bill Gates in last year, and Amadou had Bill Gates on visit last week. And so we had a 2-hour discussion on these antiviral strategies. And so the message for GVN, I think, is summarized in this slide, which we published recently in Cell Host and Microbe. So if you have a spillover event, it may lead to a local outbreak, and this local outbreak may lead to an epidemic, and sometimes, hopefully not too often, and to a pandemic. But you can only start making your vaccines at the time that you know your sequence, which was on the 10th of January 2020. And same thing for monoclonal antibodies. Well, I mean, you can think about making Panzer-Beco monoclonals, but it's a difficult exercise. Same for other families. So you can, in fact, only start making antibodies at the time that you know your enemy, but you can start making orally available small molecules just now. I mean, and when you know what the family is against which you would like to develop such drugs. And so vaccines, fantastic. They can give potential long-term protection, but there's an immunity gap and you need time to produce them. As I said, monoclonal antibodies, immediate protection, can last quite a couple of months, but they come with issues, of course. And of course, antiviral drugs also can come with issues. For example, you can, if you stop treatment, the efficacy is gone right away, and you can have resistance. But definitely, I think in the context of epidemic and pandemic preparedness, but also in the context of being able to treat infections today for which we don't have anything. Also think about Lassa and Ebola and Bunya viruses and so on. So I have some preprints for those who would like to have a copy. I can give that. Can we develop broader spectrum antivirals? Of course, remdesivir, the Gilead nucleoside analog, which was developed initially against Ebola, but was also shown to be effective against coronaviruses, is such a nucleoside analog that targets the polymerase. Same thing for the MSD compound molnupiravir. It has broader spectrum anti-RNA virus activity, not fantastically potent, but still it shows it's possible. Nirmatrelvir, Paxlovid has pan-corona potency, so it's definitely possible to have pan-family or even activity beyond families, so between families. And this is, I'm sure you know this data, with Paxlovid, and so the studies on 1,000 patients, if they would not receive Paxlovid, mortality in a certain stage was roughly 6%, and those that had been treated 0.8%. So just imagine that we had such a compound at the early days of the pandemic. By the way, as you know, Paxlovid needs to be combined with ritonavir, which complicates, certainly in multi-medicated patients, therapy. And this is something that we're trying to address. So together with Aligos, a Belgian, US-based biotech, we developed a highly potent, about 10-fold more potent than nirmatrelvir protease inhibitor, which doesn't need ritonavir. This is currently in clinical studies. Here you see some of the data. This is in the lungs, and on a log scale, the infectious titers in the lungs of hamsters. And if we treat the hamsters, there is no virus in the lungs. So, but this is all exercises that we could have done, basically, before 2020. And as I said, combinations may be needed. And just imagine that we have some viruses that may be, to some extent, sensitive to, for example, remdesivir or molnupiravir or ribavirin, not very potent, but still the compound is around. Perhaps we should start thinking about these different families for which we don't have specific inhibitors for the moment, if some of these compounds, and I just mentioned these three, if some of these can perhaps be used in combinations. None of them is very potent against some of the other infections of viruses, but maybe in combination. And this is just an example in this human epithelial cultures that we grow at the air-liquid interface. And so this is on a log scale, so we treat the infected SARS-CoV-2 infected cultures with either placebo in blue or intentionally suboptimal doses of these single compounds, and we don't see an effect. But when we combine them, we have quite a substantial protection. And the same thing in hamsters, so this is control. Each data point is the titer in the lung of hamsters. So we have these three compounds at suboptimal doses, no effect, and if you combine them, we have full protection. I'm not saying that this holds for all viruses, but it's at least something that we're going to explore if we can use the compounds that are around today to do something against some of the other families with potential epidemic pandemic potential. And so what I would like to explain now briefly is our approach. So you can have target-based approaches where you target the polymerase or the protease, but and I will explain this further on Saturday. In all these viruses and replication cycle, you have, I'm absolutely convinced, so many yet unknown druggable targets, and you need to identify them. And so the approach that we use is looking for the needle in the haystack. And so the haystack, and this animation isn't coming. Okay, so we use 384 well plates and seed molecules in each of the wells, infect with the virus, and then try to find hit compounds. And this is, for example, a screen with 100,000 compounds, and you just need this couple of hits that block completely without being toxic. Now, to be able to do this without having your technician sitting there day and night to pipe it with highly potential viruses, we, and Peter Leessen is the mastermind of the whole concept, built a lab in a box which is situated in our high biosafety lab. This is a footprint of the box. It's just 15 square meters, linked to a walk-in high biosafety lab. In the box are all the robotics. This is a glove box system that gives access to this walk-in lab. I'll show you in a minute in a short video. these are 4 incubators that can be accessed from the inside by robotics. And on the upper floor, we have all the air handling. So this is now the box. You see here 2 high-contact images with the blue lid. Here you see the door that can only be opened after gas decontamination, and all these little lids have to be opened. And now I hope that here we go. So here we grow the viruses, put it in this a transfer chamber, and now via the glove box system we can transfer this into the box. And then you have a view in the box where you see the central robotic arm. So here you see the 2 high-content images, and each instrument has its robotic arm, also the high-content readers. And so the arm is basically putting now a plate in another robotic arm into one of these 4 incubators. And in this way, for example, against SARS-CoV-2, we screened in less than a year 2 million compounds, and today we have a capacity of roughly 25,000 compounds a week that we test against, for example, rabies and paramyxoviruses and others. How much time? 2 minutes, yeah. So to end, so the example here is on dengue. So we thought that we need dengue compounds. 100 million people get dengue disease each year, and we don't have drugs, and we have it for hepatitis C. That's the same family as dengue. So we got Wellcome Trust funding and engaged with the Center for Drug Design in Leuven. And after a couple of years, J&J, Janssen Pharmaceutica, took the license on the class of compounds that we developed. And here you see these molecules. So we identified a novel mechanism of action. I will come back to that on Saturday. But this is a complex table, a long table. The only take-home message is that the activity that we have is very potent, but it's very potent against all serotypes. But if you develop drugs, you should make sure that they're just not active against lab isolates, but also against clinical isolates. So together with the lab of Xavier Damballery in Marseille, we had all genotypes of dengue, and we had activity against all of them, just to make sure that we don't miss anything if you invest in this development. I will come back to this, so it's a completely novel mechanism, and here that's basically looking for the druggable targets. This is a new druggable target in the replication cycle of the virus. And we have beautiful activity in non-human primates. This is 6 human primates infected with dengue, studies done at UTMB, and if we treat them, you have complete block of viral load. And so this compound is now in phase 2 clinical studies at J&J. But as said, we need pan-family inhibitors, and so the approach we now take is when we do the screening to do multiplex screen, and we gave dengue a blue report, the Japanese encephalitis a green one, and yellow fever a red one. Should be yellow, but red is better. And so we do then multiplex infections, and then we can basically, if we screen 1 million compounds, basically have 3 million tests instead of 1. And so that's the way we try to identify novel emitters. This publication is in press. And just to end, the Belgian government had the vision that they should invest in a new virus bank to be better prepared for new epidemics and pandemics. And this is not just a collection of viruses. You have EVA-G and the WHO BioHub, but what we basically will be doing is setting up model systems for all this family of viruses. The viruses, the reporter viruses, the cells, many cells, organoid systems, animal models have also the large libraries of drugs available so that we can start finding hit compounds, that we have a baseline basically to start using these compounds as a tool to identify novel druggable targets and replication of these viruses. And I have many, many people to thank. Thank you. I have 15 seconds left. Yeah.
Thank you, Dr. Nates. Our next speaker is Dr. Andras Wellne. Dr. Wellne is former chair of Division of Clinical Virology at the Karolinska Institute in Stockholm, Sweden, and a chief physician at the Clinical Virology Laboratory at the Karolinska Hospital. Dr. Wellne is a member of the GVN Board of Directors and its Executive Committee. He serves as a director for the Karolinska Center of the Excellence of the GVN as a co-chair of the GVN's Task Force on Long COVID-19. Dr. Valne will give us these insights about the SARS-CoV-2 herd immunity acquisition at the example of Sweden.
Thank you very much and for inviting me to give this talk. I'd probably be an enemy of Sweden, enemy of state after the talk.
Could you repeat that?
Sweden, just a little bit how Sweden is arranged for handling pandemics. We have 21 regions, and they're independent, and that provide health services. And they're responsible for the overall planning and for the health services and how they're organized. And they decide on the allocations of resources. And each region has an epidemiologist or independent infections control doctor. It's also called a regional epidemiologist. Then we have 280 municipalities, and they take care for the elderly homes. Now, interesting enough, they are not allowed to hire physicians. So they cannot hire physicians. So only the regions can hire physicians. So, which caused quite a lot of problem during the pandemic. So here's, yes, we have the parliament and the ministries. Then we have the Public Health Agency. I'll talk a little bit about that. The Public Health Agency can only give recommendations to the city councils of the regions, the infectious disease doctors in the regions, because they decide themselves what they do. And then you have the municipalities. Then we have the National Board of Health as well. And they communicate with one another. But the only ones that can really impose lockdowns or whatever is the government. But then again, the government is listening to the Public Health Agency, and they say, it's our expert agency, so we should listen to them. Now, we changed this agency, the Public Health Agency, about 10 years ago. Before that, we had the Swedish Institute for Infectious Disease Control. And it had 6 senior consultants whom also were full professors at the Karolinska Institute, and they had their research groups. And the Director General was a professor either in infectious diseases or in bacteriology or in virology. In 2009, they appointed a new Director General who was not a professor and was not a scientist. He was a civil servant. And he decided that he should decide what type of research each of the professors should do. Or if they didn't do what he said they should do, they could leave for the Karolinska Institute and leave the agency. And all of them did, of course. So now we had an expert agency without experts, but only civil servants. And now we were hit with SARS coronavirus type 2. Finland, Denmark, and Norway, very similar countries to Sweden, still have agencies like the one Sweden had before. Also, they could make recommendations to their governments. But then again, they had their experts. And so the governments did listen to them. Mind you, they started in Denmark and in Norway, more or less the same way we did in Sweden. There was a slack attitude. But the governments interfered very, very quickly in Denmark and in Norway. And so Denmark and Norway did very well. Finland was very active right away. So here we had an example of politicians who actually took and and realized what had to be done. Not so in Sweden. And I can tell you, the government was listening to this so-called expert agency, but so did also the opponents, the opposition parties. So we couldn't get them. They said, we have our expert agency, we have to listen to them. And it was a little bit like, you couldn't criticize. It was like as if Sweden had been at war. And if you are at war, and then everybody should gather, and everybody should, just do exactly what people said you to do. Anyway, in 1940 also, what happened was that previously we had the Infectious Disease Control Agency, and they only did recommendations on infections. But now they said that this public health agency should have a broader thing. They should look at health at large. So whatever they did, if they implemented lockdowns and so forth, what would that do to for instance, depressions? What would it do to money-wise? Would that, and so forth, which are really political things. So they didn't recommend on how to handle the SARS-CoV-2 infection when it came to Sweden. And remember, it was in Italy 3 weeks before Sweden, and people were dying in the Italian Alps and in Austrian Alps. And we saw it, and we saw how they were treated. They were, on average, 11 days in intensive care units, and many died. Now, the Swedes that were infected came back to Sweden and just entered the country. There was no control of those people at all. And they could start work the next day. They could go to school the next day. They weren't even asked to maybe stay at home for a week. So, what did we know in the early February about this disease? Well, we know quite a lot. There were only 200 papers. I read them all. But it was absolutely obvious that there was a pre-symptomatic and an asymptomatic virus shedding. So that children got infected, and secret, there was a child, for instance, from Singapore, who was taken care of in the hospital, and they tested her every day because she had no other care to care for her, and both her parents were in the hospital. And she excreted more virus than both her parents together. And of course, she was infectious. It was also very obvious that this was an airborne disease. And we knew that from earlier, from cow coronavirus, which is airborne disease. So why shouldn't this be? And of course, face masks should be of value, even if you couldn't pretend getting infected, you would reduce the infectious dose. And the ones of us who's been doing experiments with animals and infecting animals, we know that the dose is so important. And if we can reduce the dose, probably people wouldn't be that sick. I know for some viruses that doesn't work. Anyway, so the Swedes had this very lax attitude. So they decided on mitigation rather than suppression. And some of them had been signing the Great Barrington Declaration, and which was the declarations where protect the elderly, and but let it spread amongst everybody else. It's just that they never thought that the elderly needed care. And that care had to be provided by younger, who then would be infected. So the younger providing care would infect the elderly. That's exactly what happened. Mind you, the loved ones were not allowed to see the elderly. And this happened in Sweden. So there was a press conference every day. And they denied every day that there was pre-symptomatic transmission. So if you feel fine, go to the job. You won't spread the virus. And same thing with asymptomatic transmission. And they said that if you keep 5 feet from other people, then you're fine. You can't be infected. And they really discourage use of face masks. And they said that it probably will not prevent, but on the other hand, you will get more infections if you use face masks, because you get the virus on your hands. And so. Then again, I mean, it was very early we realized that this was not a virus that was infected by food mites. But then again, they couldn't say that they were not allowed to say that they're going for herd immunity. But in between, they said that they didn't want to stop the spread of the virus infection totally. They wanted it to spread so that people would get, immune. And during the spring of 2020, we had these calculations by mathematicians that we would, by June, have like 50% of the population being infected and immune. So they said that let's mitigate, and you've probably seen this cartoon. Let's mitigate so that we don't have so many, so that it will overwhelm the healthcare system. So try to keep patients here under the curve. And how do you do that? It's impossible, I can tell you. And here is just the Director General saying that children don't need to stay home because they don't spread the virus. Situation in March 3rd, we had 90 cases, and more than half of them were infected in Italy. And we had a very, very poor test capacity in Sweden, and reagents were gone. A few days later, then state epidemiologists said that we'll get a few more cases and then it will be over. And mind you, just 1 month later, we had the most deaths from COVID-19 in the world per capita. Over 100 persons died every day in COVID-19. And by June 18th, 500,000 had been infected. It's 5% of the Swedish population. And 5,000 had deceased, which means it's a mortality of 1%. Now we calculated, okay, here are the messages that came, the same thing as that. We calculated how long would it take to reach herd immunity. We have 500 beds in Sweden, and we calculate that you needed 70% being infected in order to get herd immunity. So, and we have a 1% case fatality rate, and at least 2% needed intensive care units. And we know that the average time was 11 days. So with a population of 10.45 million people, that would mean 7,032,000,000 days in intensive care. Million. And for 11 days, that's 1.6 million intensive care days. And you divide that by 500 beds, and you get 3,200 days, and then you divide that by 365 days a year, and you see you're up to 8.8 years. Thus, it would take almost 9 years to reach herd immunity in Sweden by letting the virus spread without overwhelming the healthcare capacity. Now, and this is calculated that each bed in the ICUs was used for coronavirus patients, for COVID-19 patients, not for open heart surgery, not for anything else, which of course needed. We tried to set up new ICU wards, but the problem was that we didn't have qualified personnel, so we never used them. So how did we cope with this then in Sweden? We had all these patients. Well, the elderly patients were not to see doctors because there were no doctors in the elderly cares. But they were given oxygen, which is contraindicated if you have breathing problem. So, morphine, yes. No, I said they did not get oxygen, but they were given morphine, yes. So they killed them. Or if they were agitated, they would get oxazepam, which is indeed contraindicated together with morphine. So if they didn't die from the morphine, they certainly died from the oxazepam. They call it palliative treatment, but in fact, I would say the people were killed. The elderly were killed. Now, in many of the regions, they said that if you were biologically over 57, they couldn't say calendar age, but biological age. If you were over, you could not attend a hospital. But then again, if you're deadly sick and you're unconscious, how can you tell the biological age of that person? It's impossible. So, by definition, everybody who came in deadly sick had a biological age over 75 and were treated as such with palliative care. More time? Yeah. Here is a graph which I think tells everything. You can see these are the green one are the ICU cases in Sweden and their age. So this is 80 to 89, 70 to 79, and so forth. And here are the ones that died. And you can see that the ones that died were not treated in the ICU care. So, yeah, I'll have a few slides on we did so much worse than our other countries, which are very, very similar. I'm just show you this last slide then. This was until the end of 21, and you can see the cumulative number of deaths in COVID: Sweden, Denmark, Finland, and Norway. This is not something to brag about. I think this is probably the saddest time in Swedish healthcare, these 3 years. And I'm ashamed. And we would have needed the Global Virus Network to come into Sweden and say, you can't do this.
We should.
We wrote more than 100 opinion letters actually one that is still the most read opinion letter ever in Sweden.
And the government survived?
Sorry?
Did the government survive to the COVID-19?
No, it did not. And the state epidemiologist did not survive. They said he got a very, very good job at the WHO in Geneva. So he left. But the problem was that they forgot to tell the WHO in Geneva that he got it. So he was actually fired. Yeah.
This is really could be the most important topic of India.
Yeah, but it's important. It's really important. Okay. Yes, absolutely.
Okay, and we came to the last talk of today's session. Our next guest is Pascal Barbry, Research Director at the French National Cancer Center of Scientific Research, CNRS. He holds the chair for Artificial Intelligence for Computational Biology Data and Bio-Inspired Artificial Intelligence at the Institute 3 AI Côte d'Azur since 2020. Dr. Barbry is the creator and head of Physiological Genomic Group and the UCA Genomics Platform, coordinator of the Academy Complexity of Life at the same university, scientific leader of the Equipex Plus 4D Omics, founding member and member of the Executive Board of National Infrastructure France Genomics, president of the Canceropole Provence-Alpes Côte d'Azur, and president of Scientific Council of Association Vaincre la Mucoviscidose. Dr. Barbry will talk about wastewater surveillance for infection disease.
Thank you very much. Thank you also to the organizer for the invitation, for the kind invitation. And so I think that I will come back to a less polemic aspect of the of the discussion by discussing about our own experience with wastewater. I saw already several presentations about that, and I will share with you the results that we have collected in Nice, in the Nice area. And I have to say that I am not a virologist, and that was the organization that took place at the moment of the pandemic, when we were facing a lot of difficult situations, especially because nobody was really sure to be able to test what has to be tested. And so we tried to think about what we could do with our expertise in the domain of the sequencing. And so the idea that was already introduced was to try to consider, rather than the sequencing of individuals, a more global picture that would give a picture, a large picture, an epidemiological picture that can be then explored and used by the health community. So, by comparison with the sequencing of individuals that provide information about novel mutations, or that allows us to track the spread of mutation from one person to the other, this situation is counterbalanced by the high cost and time that is needed for screening large populations. And having in hand the possibility to do just one sample representative of a whole large population would be interesting to develop more comprehensive testing of this population at probably a cost-effective in a cost-effective situation. The drawback of that approach is that in a single strain, you know that you will exactly define one lineage when you are analyzing one patient sample. While in the case of the multiple strains that you have in a complex sample, you know that we will have you will have a mixture of lineage, and this is something that you have to consider. So this is why we rapidly switched to feces and wastewater. And at that time, there were already a lot of reports suggesting that it would be possible to do so. And so we went to the municipality of Nice, and we asked whether we could have access to their wastewater system. Nice is interesting because it is a metropole with a quite large population. There are a lot of tourism, and so you can imagine the exchange that can be at the viral level. And on top of that, there was a well-organized sewage service. So I have a different view to share with you about the Promenade des Anglais, which seems to be beautiful there, but in fact, you can consider it just as a very large pipe going from the city to Aliotis that correspond to the wastewater treatment plant. And so if you consider that large pipe, you have some perpendicular pipes that are directly going into the different areas of the city. And so with the wastewater services, it was easy to organize the collection at the different levels and to have by this way a picture in the different area of the city of what was going on. And this is something that we have done during the early of the end of 20 and the beginning of 21. We collected about 100 samples and we sequenced them using PCR amplification with Arctic sets of primers. At that time, the situation was quite clear, and with a small number of mutations, it was possible to identify the different variants. And you see that, for instance, from October to January, the city of Nice was mostly covered with B160 variants. And there was a sudden shift at Christmas of 20. And this is, by the way, the arrival on the Promenade des Anglais of the English variant. In fact, what is interesting is that it came not immediately and uniformly in all the city, but you see that we have a spot, an area in the city that corresponds to a specific neighborhood of the city, where it developed particularly well. And what is interesting is that in parallel to what happens in January, so around Christmas, there was also other variants that were detected in different areas of the city. But this is this one that took over and that win the competition with the other variants. And so what is interesting from that perspective is that we can have a picture of what's going on, and what is particularly noteworthy is that this particular area where different type of variants were developing were specific areas, specific neighborhood with lower income, poorness, higher density of population, as it is usual for this sort of situation. But we have directly a picture of that. And the second lesson that we learned from it, from this first experiment, was that when you collect all the information from the different area, you have a good picture when you compare what's going on directly in Aliotis, the central wastewater treatment plant, or when you are doing the average. And so based on that, we shifted the protocol, and we decided to do only one sample each week and to concentrate only on the wastewater plant. The problem at that time was just to modify a little bit our protocol so that we can have a sufficient complexity in the samples that we are analyzing. And the drawback, the problem that is that people are facing when dealing with this type of sample is that you are not only purifying RNA, but also a lot of inhibitors, of enzymatic inhibitors. And so we changed the protocol, and we set up a protocol that was at the end particularly useful, and that was able to work for 200 milliliters in routine, but with a very wide range of possibilities at the end. And we added some specific primers to analyze more precisely the situation in the spike to ensure a good coverage in critical regions of the virus sequences. With that, with all this improvement, we improve the coverage, we improve the percentage of mapped reads, and we improve the typing. For instance, you see that we have nearly 100% of coverage in the area of the spike. And by the way, we have compared the protocol with the protocol that was proposed at the national level by SUMO, the national network system. And it turned out that the protocols that we have set up at IPMC gave us the highest sequencing coverage, the highest mapping percentage, and was highly reproducible to identify small variants. What is also interesting, and that was a question that was asked, is that technique really quantitative? We have been able, in collaboration with Santé Publique France, to compare the percentage of the different variants at different times of collections. And we found a very good parallel between the flash sequencing of patients and the analysis of the wastewater. So we are really convinced that you can get a direct picture of what's going on in an area by working just with wastewater. And we have in parallel set up digital PCR, and it is also useful for that. So the advantage that we found with that was that the sensitivity that we can attain with sequencing can be particularly high. And I will just illustrate that with some examples. The first one is the arrival of the Omicron at the end of December, in December 2021. And at that time, Omicron is there. It represents less than 1% of the total number of reads. But it was possible to identify it among the different reads just because we found a few reads that containing the very specific pattern profile that was found around the spike and that was characteristic of the Omicron. Based on that, we probably profiled the Omicron when there was less than 20 people positive for this variant in the population of Nice at that time. And in fact, we did the same afterwards to identify some recombinant variant like the XBB, also with the XBB1. And more recently, we were able to identify the Pyrola. The Pyrola was interesting because we found, again, a small number of reads that represent only less than 1% of the total reads. And we found it in the population in one sample, but we were not able to detect it before, and it never came after. So probably we were able to catch it, but the competition between Pyrola and the other variants was not good for Pyrola, and he was not selected in the population. So we are now moving to a different protocol because we think that it is important to set up now a new survey, especially for a larger population of viruses. And so I will just show you to the end of my presentation the first results that we have obtained with a viral surveillance panel that is proposed by Illumina. That, by the way, was sequenced with nanopore sequencer. And in that case, it was possible to, we have profiled just about 10 samples from 22 and 23. And we identified 29 different viruses into our samples. Fortunately, the 37 were very dangerous ones, and so we didn't get them, and that's a good thing. But what is interesting is that you can have an idea of the different time course of expression over the time. And you see that for some viruses, like astroviruses, you have a very stable expression, while you can detect some spike, some peaks of expression at different time of the year for some of the viruses. So, for instance, we detected some polioviruses in the middle of 22, and it seems that it came back in 23. We were also able to detect the SARS-CoV-2, and the profile were very close to the ones that we have obtained before. There were also the expression of other coronavirus, and what was interesting also was that it was possible to detect, though at a low level, some influenza viruses, and different strains of influenza viruses were not observed exactly at the same time. So this is something that we will document better now. So in conclusion, I think that I have shared some evidence that wastewater sequencing is becoming a really credible solution for viral epidemiology. We demonstrate its value at a scale of a metropole like Nice, with its 400,000 inhabitants. There are specific procedures that are necessary for preparing the sample, collecting it, sequencing, in order to ensure a sufficient complexity of the sample for variant quantification. That implies a sufficient coverage of the genome, and also a very large effort with regard to the bioinformatic resources in order to be able to correctly identify the circulating variants. And just to finish, I would say that our implementation allowed us to alert very early the local authority when alpha variants, Omicron or Pirola were coming, well before their detection by the health system. And so finally, the approach is also applicable to other viruses. And to finish, I just want to thank my collaborators, especially Rainer Wallmann, Geraldine Rios, Pauline Station, Sarah Herfi, Anna Diamant, and we have also a nice collaboration with Benoît Gassillou and Sumo at the ANSES, and Frédéric Jourdain at SPF, and we are linked to the OBEPIN network, which is a research network for viral detection in wastewater in France. Thank you for your attention.
Thank you, Dr. Rabi. So now we will start with a roundtable discussion, and for that I will invite President of the General Virus Network, Dr. Christian Bréchot, as well as Drs. Marianne Kupperman, Dr. Nates, Dr. Wang, and Dr. Wellne to join me here on the stage.
Well, thank you very much to Melissa Wojciech for chairing the session, because it's a kind of miracle we've finished on time. You did a great job. Thank you, and thank you so much for the experts. And so we have some time for some questions before getting drinks. So who wants to ask a question? Bob, you wanted to ask a question. Yeah, no, it's coming. It's coming.
Yeah, well, I had a question before for Johan, then you have one for Anders. Anders is very quick and easy, and everybody remembers the talk so well. So I start with Anders. Now that you changed the government, did they change and go back to the policy of having the virologists at the infectious disease people at Karolinska doing the advice, or did they keep the civil servant, as you call the poor fellow or woman?
No, they are considering going back.
Considering?
Considering. They haven't done it yet. It's they're very slow. You know, they weren't in opposition to the government while it lasted. So we talked to the opposition party leaders, and they said also that we have to listen to our expert agency. And we said there are no experts at that agency. So
yeah, but it's inherently obvious from the data you presented,
they
have it in their face, and they have
but right now hands. They are looking into it, and most probably it will go back to as it were before.
And Marianne wants to. Marianne will address this.
Yeah, I cannot comment on, well, I know the reorganizations happened in many public health systems actually in Europe. I'm a little bit uncomfortable with the, pointing out one person. We've seen scientists being threatened, so I think we have to be careful that what in the statistics, if you look at the rate of mortality, so mortality per million, there's a whole list of countries higher than Sweden. That includes several countries in Europe, France, Italy. It includes the US, it includes the UK, it includes Belgium. So many countries. So I think it's really important to do the evaluations, but also be a bit careful about it. I understand your...
But then again, this may be a conversation for the drink, actually.
Yeah. No, it is important.
I don't think you
can make that comparison.
That's why we compared with the Scandinavian countries. They're about the same density. I mean, if you take Belgium, for instance, there are about, I think, 30 times as many people per square mile. Sure. And Monaco has got... Sweden has got 24 people per square kilometer.
There's no comparison.
Monaco's got 24,000. And I think England has got like 300 something.
But it's not just that, Anders. Wait a minute. You jumped right to Italy. I mean, Italy got wiped out at one point in time. And that's the North Italy. Well, no, wait, let me finish this point. They had 200,000 Chinese visitors that went back for the Moon Festival or whatever it was. And they got a lot of people infected who came back that were working in Milan area for the fashion industry. Sweden doesn't face that. No.
But I mean, if we compare, I mean, Denmark is much more densely populated than Sweden, but they did the right thing.
Okay.
And Norway is just as densely populated, and Finland, and they did the right thing, and we did not. And what really pissed me off was the disinformation that they gave, because not even today will they say that it's an airborne disease.
Yeah. That it's what?
An airborne disease.
Yeah, okay. Well, the United States had a visible and friend that was also smart, and he was taking the position that there was no aerosol transmission, that we kind of early on didn't really need the masks. And
we know so many instances.
I think that if we start to analyze all
the asymptomatic
all the mistakes worldwide, I think that we are not going to go through this roundtable. But I understand your point, Marion, and it's important.
I think if the GVN wants to take an advisory role, it is really important to dive into the details and say, can we understand what is happening here? And for instance, airborne was a very complex discussion. So clearly it was airborne, but airborne in terms of public health, what you put in place is very different things. If you say you can do a lot of things with just physical distancing versus you have to consider total airborne, very widespread, and those are very different things. So.
Yeah, but then again, killing people is never right, and giving people who can't breathe morphine is never right.
We will move to other questions.
And one last point. And the asymptomatic people. Just let me finish the point. The asymptomatic people was also stated in the United States, and until you have clear evidence that asymptomatic don't transmit, you shouldn't say it. That's obvious, and it's common sense, okay?
What I take from this conversation, and this is really for Sten also, is that we may still need exactly what you said, Marion, to try to have a common position on some of the stances which were taken during this pandemic. No, thank you, that's very useful. So we have several questions. We have Mark. Sorry, please go. Sure. And then Mark.
I wanted to say thanks to be here. I am a human geneticist working in Naples in Italy. And from all this conversation, I would like to suggest to take in consideration how the genome, the human genome, is dealing with the viruses, and as well as the animals. I would say all the spillover studies and the Singapore approach, I think it will be really benefit of several studies just showing potential of impairment of the ingress of the viruses in the human genome, or having the asymptomatic type of geneticist environment, just to have that information to make maybe some drug repositioning, or trying to verify if there is a way of fighting new viruses by using the human genome genetics.
So what do
Can you
think?
comment on that?
Maybe Linfa, and then maybe...
I mean, clearly, you know, this is a precision medicine. So in Singapore, I am the ED of the PREPARE program, and there's another multi-million-dollar program called the precision medicine. So Singapore is 5.8 million, and we try to get 100,000 genome, basically, regardless of your health status. And these two programs, of course, we're talking. But I think your dream scenario is try to use the human genome information during a pandemic to try to, you know, help. I think feasible, if possible, but currently I think the technology may not be there yet. So Singapore is doing that. The precision genome project is mainly driven by cancer, chronic, slow kind of disease process. For the acute disease, we try to plug into this. The difficulty now is the AI, the talk about AI versus privacy. That is the biggest barrier right now, because these are so detailed data of individuals in Singapore. In terms of disease prevention and understanding, it's very useful, but in terms of privacy, it's a nightmare. So even in Singapore, we're still struggling, how to handle that.
Yeah, that's right. Thank you. You have a point?
Yeah, and perhaps just, I mean, so I have two aspects. I think it's blocking viral replication. I think we have to do that by directly acting antivirals. And then perhaps somehow along your question, I mean, disease-modifying compounds. But that's not my field. But from what we learned also this morning in the AI talk, I mean, this is definitely, I think, a path forward there. Just one example is that we found, and this was just by kind of coincidence, we found that nelfinavir, the old anti-HIV protease inhibitor, that this inhibits in cell culture SARS-CoV-2. So we thought, wow, this was early on in 2020, and so we tested that in the hamster model, and there was no effect whatsoever on replication of SARS-CoV-2 in the lungs of the hamsters. But for some reason, we had sent his lungs anyway to the pathologist, and his lungs were clean. So what we found is basically that nelfinavir, by having an impact on neutrophils, is blocking basically disease evolution. And that's perhaps, I think there is some room, I think, for compounds that are disease modifying without having an impact on replication.
Thank you.
Yeah. Okay, I am.
Marc Bonneville.
No,
but Marc and then Jean-Paul. Marc.
Okay, I found very, very interesting the talks of three of you regarding wastewater surveillance, and I was wondering, typically, because Pascal and Marion clearly showed how sensitive could the approach be, and its efficiency in terms of detection of novel viruses or cryptic viruses and so. But I was wondering what could be the organization actually of the approach in order to make it feasible and scalable. Typically, Pascal, you described this very nice approach implemented at the level of a city. So do you think that it would be implementable, for instance, on a national basis? And or do you think that you would have actually to organize this in such a way that you would have actually a very limited analysis of a small number of samples by metagenomics and switch very rapidly to either targeted PCR or whatsoever?
So I thank you for this question. So the idea in France, developed by Aubépine, for instance, was to have the largest number of sampling stations. They have over 100 or 100, nearly 150 at the moment, but they were doing only quantitative PCR. And so the strategies that we have chosen was quite different because we focused on one spot and we decided to look in that condition what was possible. We have compared some sample coming from different areas of France, even from Europe, and at the same time point during the pandemics, there was well, it was not exactly the same, but for instance, in France, I think that the shuffling of the variants in the population was quite rapid, and the variation from one point to the other was not that high. So I suggest, this is my idea, this is not shared necessarily by everybody, but I think that if we select some, let's say, 5 to 10 big cities in France, highly connected because airport and things like that, you can probably have a good glimpse of what's going on. And then this was more on political issues because, well, mayor of cities wanted to say that they have done something, so they wanted absolutely to get the value for that city. But I'm not sure that it was really informative. And if we do that cleverly, I really believe that even for France, about 10 to 20 spots could be sufficient to have a good glimpse of what's going on. But I'm not an epidemiologist, so...
No, but I agree, and it's the same approach the Netherlands is taking, and I think there's a European JRC-led consortium that looks at this in many countries. I do see, so I think that is the way to go for monitoring what we know is there and maybe variants thereof. I do think we can actually do some deeper dives with metagenomics in high-risk sites. So, for instance, that could be added to the airport wastewater, for instance, or to slaughterhouses if you want to be able to detect other things. But the metagenomics really is still exploratory, but we find, for instance, viruses that tell you there are Aedes albopictus mosquitoes in there. You know, that's an interesting observation. You could actually then think about using it for very different types of surveillance questions.
We have, in parallel, done some measurements on the Nice airport. By the way, it's a very important airport. But, well, usually the value were not as reproducible as in haliotis, just because the sampling was not optimized. And I think that there is certainly a very interesting work to be done with the airports to collect carefully the information. We are at the opposite of the usual way. The information that you can collect are for people that are leaving the territory, and you can inform the countries where these people are going about the type of things that they have. And this is at the opposite of what people want to organize. When they want to organize something, this is for themselves, and they want to know exactly what will arrive on the territory. But the problem is that when people are arriving, they can use the toilets. That is interesting. You can collect some information. But immediately after, they are leaving the place, and you cannot do anything from that.
It's too late. Yeah. Jean-Paul Gonzalez.
So we want all save life and for the people, and that's what we are here. So treatment and vaccine are our tools. We are using, that's for you, yeah, we are using since the Pasteur area and Koch area, we are using polyclonal antibodies. Why are you, it looks like we are forgetting about polyclonal antibodies, switching for monoclonal antibodies, which are not so efficient. What's your opinion about that? Polyclonal antibody versus monoclonal antibodies.
Well, what?
I think the results were good.
Yeah, no, I mean, has been quite a bit of studies with convalescent serum that for SARS-CoV-2 that showed that this was not working, almost not working, I think the production, the biotechnological production of monoclonal antibodies, I think that's somehow the way to go. I can't see really an efficient way of producing polyclonal antibodies in an large epidemic, pandemic situation. That's
Thank you. We have two other
the elephant
questions.
farm. Or,
No, we have three
Buffalo.
other questions. Please.
I agree with Marion that metagenomic analysis is really exploratory, but I think it's a way of the future because there is no other way you can have this broadly specific detection. And my question is actually to Pascal Barby. Was there anything in your analysis that was really unexpected, that you raised your red flags? Because for me personally, I think you've mentioned on one of your slides that there was polio that you discovered in Nice. I mean, did you dig further into it? Is it a vaccine virus or is it a wild? Did it raise any concerns?
There are some variations, and well, the natural history of the virus was not exactly as so we saw some point mutations that differ between the reference variants that we were looking for and the ones that we were detecting in Nice, for instance. But we have not dig too far into that. There are some literature about that with the discovery of some cryptic mutation in the spike regions, and that could be of interest. But we are not looking at that in the detail so far.
To me, the polio is an example of the power of wastewater, where we have seen expansion of polio in London, and that led to enhanced vaccination without a human case reported, just because of wastewater positivity. So I think that's a very nice example of what you could potentially do.
Yeah, very convincing, yeah? Thank you. We have Richard Sherman, and then we have you. Sorry? We have so many questions, which is great. But, and I have the light you see here, so I hardly see those on the back, so I apologize.
One comment related to the Andres and Marion. You know, the excess of mortality is very important from the pandemic, and this is one of the goals to follow up. I remember because Peru was one of the highest mortality with the pandemic, but the main reason is because the excess of mortality was created as the mortality from COVID. And real 1/4 of these people who die is dying because they have not health system. They have dying from meningitis, from diabetes, from HIV, etc. And we're very important when we're asking what happened in Sweden, because this excess of mortality is very important. For instance, New York and Moscow, they have the highest mortality in any city. You review the excess of mortality they have. And again, this is important when you mention about the Sweden. It will be very important to understand how many people die from the COVID and how many people die from the loss of the health system. That happened.
I know you're asking how many died with COVID and how many died from COVID. The ones that we...
No, not really. He's really, as I understand, he's really making the case that to really interpret the rate of death, you really need to compare with the rate of death which are not related to COVID-19, be directly or indirectly. I mean, the kind of baseline, which in fact in Sweden is likely to be low.
So the ones that in the official statistics, they died from COVID. That was decided. Now, if you look at excess death rates, because that has been something, then you have to calculate what should be the normal death rate. And in Sweden, the death rate has gone down all the time.
That's the answer, actually.
And whereas in Finland, it's been going up because they have a very elderly population. Remember, in Sweden, 20% of the population wasn't born in the country, and they came into the country, younger people. So if you extrapolate the line you had before, and then take the line for 2021, 23, you get 24,000 extra dead, and which is exactly, more or less exactly what died from COVID-19.
Thank you.
But the apologetics, they want to make, they don't take into account that we have a downs, the trend is going down in Sweden. So they just did a medium of the 3 years before, and then they half the excess rate. So
okay. So now Richard, and then we have Bob Gary, and we have you. We have three.
Okay, Bob Gary from the GVN Center at Tulane University in New Orleans. I have a question for Linfa. Linfa, I noticed on your pre-pandemic slide that you had a lot of neutralization of RATG13.
Yes.
Now, I know you don't want us to tell us the countries, but do you think that's spillover from bats? Because RATG13 is obviously a bat virus. Or do we have an unknown Sarbeca virus that's circulating in people?
I think more than just bats. I think the bat-to-human spillover is actually very rare. It's more the small mammals. So that, I mean, COVID-19 is a good example because we know the ancient virus, the ancestor virus is in bats, but the progenitor virus, I can almost say it's not from bats. So the outbreak was caused by a virus from animal, and most likely the trading was involved. But, so the bat virus actually is not the progenitor virus called SARS-CoV-2. So there are Sarbecoviruses circulating in small animals all over the world, and that spillover is happening all the way from very north to very south. Yeah, so RATG13, we actually, we have identified RATG13-like viruses, which are much closer to SARS-CoV-2 now, and they use ACE2 receptor much better. So lots of viruses out there, and the spillover, as I have shown this data, is happening more frequently than we thought. Yeah. I mean, Singapore, we have the T-cell data. Even in Singapore, such a small, clean island population that, Antonio Bertolatti, did the T-cell. I was involved in the study. We have a COVID-19 cohort survived in May 2020, and the SARS-CoV-1 survivor, we had, infected in 2003, and we have 50 what we call healthy, neither SARS-CoV-1, SARS-CoV-2. When you do their T-cell, really specific T-cell assays, that 50% of the healthy already have specific T-cell against both SARS-1 and SARS-2 in Singapore. Yeah. So basically, in our human population, we have been, seeing related virus for a long, long time.
Yeah, those pre-pandemic samples are so valuable, and I think that's one area where GVN centers can help, because there are probably samples in people's freezers that will be very revealing. So,
Thank you very much. Richard?
Yeah, this is also a question for Lifan. So I think you made a really nice point about the importance of serological surveillance. That's very complementary to the genomic surveillance. And also that we've used neutralizing antibody as a pretty good surrogate of protection historically. But our group and several other groups have been working on pan-coronaviruses, and we're seeing that there are formulations that are giving us protection in different models without neutralizing antibody.
Sure.
And so I'm wondering, are you seeing the same thing? Is this related to the T-cell response that you were just discussing? And I'm curious if you're observing something.
Yeah, certainly. As a correlative protection, I think, much more than just neutralizing antibody. But I keep saying neutral antibody is very important if we want to prevent transmission, because this is the first line defense, basically. Okay, that's point number one. Point number two is I'm not using neutralizing antibody here in my presentation is not as a correlative protection, as the best tool we have to have the specificity in serology to declare that Sarbecovirus spillover happens in human. Because if you use a binding antibody, then people say, oh, it's OC43, or it's 229E, because we know have the seasonal coronavirus happening all the time. We can assure at least that's not that. It's spillover, but we don't know what kind of virus and which animal caused that spillover.
Thank you. And I think behind you, yeah, but
we also have,
and then, of course, but please.
Okay, thanks. Speaking about antiviral drugs, is it possible to activate PGP protein if we use it for a long time? And if yes, is there any issue about it?
I wouldn't do it. I think if we just think about for the future developing novel drugs, I mean, you just have to make sure that the PK of these drugs is okay, and I wouldn't start playing around with modulating host factors. I mean, just think about all the HIV drugs or hepatitis C drugs. I mean, you can just do it clean. I wouldn't play tricks on these things.
Okay.
Yeah.
He doesn't like it. And then the...
Okay, thank you very much.
Maybe the final question.
Thank you very much. Mine is not too much of a question, but just a concern, because I know somebody will be talking on Lassa fever tomorrow about Gary. But because the issue of antivirals were mentioned, I thought that and Lassa fever was not mentioned among those drugs that are that is being used currently. So I'm wondering, apart from the ribavirin that is currently in use, do we have other drug that is any update from the antivirus?
Yeah, that's an important question. I think we need indeed drugs to treat Lassa and also look at arenaviruses in a broader sense. Favipiravir, the flu compound, has some activity against arenaviruses, but there is really not much. It's and something is really needed there. That's my belief. I don't know, Bob, if you agree. Yeah.
Thank you. Please.
Yeah,
you will talk. Okay, well, that will conclude the roundtable. Thank you so much. I mean, really very good discussions and presentations.